Hello! In this RLabs tutorial, we will produce a simulation model of genetic drift in R. We will do so using functions such as RUnif and RBinome, which we saw in the accompanying stochastic model video. We will also use while loops and for loops. And if you're not familiar with these functions, you can refer to our lesson specifically dedicated to for loops and while loops. So first we have to understand that genetic drift is simply the occurrence of changes in gene frequencies caused by entirely random events, so not by selection. For example, let's consider an asexually reproducing population of constant size n. In this case, n is 10, containing two types of individuals defined by two different allelic forms of a particular gene. We'll call these forms G and B in this example, represented by green and blue individual squares respectively. And here we assume that the two types do not differ in their likelihood of survival or reproduction, so there is no selection. Now in any given generation, let the proportion of G and B in the population be PG and PB respectively. Now consider how the proportions of type G and type B individuals might change from generation to generation due to chance events. Well, one way to proceed, and the way that we'll simulate here, is to simply assume that each individual in the next generation has a probability PG of being type G and a converse probability PB of being type B. So each and every individual in the second generation, which we know has the same population size as generation 1, has a 50% chance of being green and a 50% chance of being blue. Now we might expect, given these odds, that generation 2 would comprise 5 green individuals and 5 blue individuals like generation 1, but due to chance alone, generation 2 might look like this, with 3 green individuals and 7 blue individuals. So the new proportions of green and blue in the population are 0.3 and 0.7 respectively. So now in generation 3, each individual has a lower probability of being green than blue. So now, due to chance, generation 3 might have this distribution, with only 1 green individual and 9 blue individuals. Now again, by chance alone, the number of green individuals could have gone back up in generation 3 and can still go back up in generation 4, but just to keep this example short, with such a low probability of each individual becoming green in the next generation, let's just assume that generation 4 ends up consisting of only blue individuals. So now we have fixation. The green individuals have gone extinct. So now let's simulate something similar in R. Let's assume that we have two types, A and B, that have equal fitness and reproduce asexually in discrete generations. The total population size, n, will be 50 and will remain constant over time. In the first generation, there will be 25 individuals of each type. Now obviously, as we know, in the next generation, each and every one of the n number of offspring will have probability PA of being type A and PB of being type B. And we want to observe how the population changes in composition over time until either type A or type B goes extinct. Now there are a couple of ways to simulate this in R. The first method that we will see uses the RUnif function. First of all, we will specify a population size n, which we said was going to remain constant at 50. Next, we make a vector to store the changing proportions of type A individuals throughout the generations. Now note that we will only work with type A individuals here because we can figure out the number of type B individuals with the total population size n along with the number of A individuals. Likewise, we can determine the proportion of type B individuals simply by subtracting the proportion of type A individuals from 1. So we really don't need to explicitly refer to type B in this code. But I just arbitrarily chose to work with type A. I could have just as easily worked with type B instead, but we only need one of the two types. So here we have the empty vector PA of undefined length. Now we know that in the first generation, 50% of individuals are type A, therefore the first element of the PA vector should be 0.5. Now since we will be calculating the proportion of type A individuals in multiple generations, we will need a counter to keep track of each generation we're dealing with. And that is why we have this index variable I. It's set to equal 1 initially since we will start by considering the first generation. Then we will continuously increment this I value within the while loop that you will see soon so that we can refer to other generations aside from generation 1. Now here is the while loop that will allow us to calculate the various proportions of type A over multiple generations. Essentially, we don't know exactly over how many generations we will need to calculate these proportions. We just know that we will have to keep track of the proportions until either type A becomes extinct or type B becomes extinct. So that is why we will run the loop while any given element of the PA vector, the proportions vector, is less than 1 and greater than 0. So just to reiterate, this while loop controls the number of generations over which we will be calculating proportions. Everything within the while loop happens once for every generation until fixation. Next, we have a variable called NA, which represents the number of A individuals. It is set to equal 0 right now because this is acting as a counter of the type A individuals that will be in the next generation. This for loop below is going to determine whether or not each individual in a given generation is type A, and then will increment the NA vector accordingly. The next part of the code that we will look at is this for loop here. It says for j in 1 to n, which means that all of this code in here is going to be executed 50 times in a row for any one given iteration of the while loop, so for every generation. Now why 50 times? Well, once for each member of the population, which is always of size n. So for each member of the population, we're going to generate a single random number between 0 and 1 using the RUnif function. Then we use this if statement and say, if that random number is less than the given element of the PA vector, then add 1 to the NA counter. So this if statement is used to determine whether or not each member of the population is type A. Essentially, we can think of this as a number line that ranges from 0 to 1. Some of the numbers on this line represent type A, and the others represent type B. So if there is an equal proportion of type A and B individuals in the population, then a random number between 0 and 1 should have an equal chance of representing type A individual and representing a type B individual. If the proportion of type A individuals in the population is greater than the proportion of type B individuals in a population, then a randomly generated number should have an equally greater chance of representing type A than type B. Likewise, if the proportion of type A individuals is less than the proportion of type B individuals, then a randomly generated number should have an equally lower chance of representing type A than type B. Now, since this for loop will do this for each member of the population, once the entire for loop is done for any given generation, we will have a total number of type A individuals for the next generation. And so we can store that value as a proportion as the next element of the PA vector. And then we can increment I, thus moving on to the next generation to do the exact same thing. Now let's execute this code in R and see what happens. So here I have executed the code that we just saw, and now I'm going to plot out the results. And there we go. We can see the changes that type A experiences through generations. So that's all for the first method, the RUnif method, and now let's look at how you can achieve the same end result using the RBinome function instead of RUnif. In my opinion, this is actually the simpler of the two methods. So here's the bulk of the code, and you can see that it's very similar to the other code. We set the population size, we make an empty vector to store the proportions of type A, we set the first proportion to 0.5 since 50% of individuals in the first generation are type A, and then we have our index counter again, I, which is set to equal 1 initially. Now here's where the code starts to deviate from the first example a little bit. We still want to find the proportions of type A for multiple generations, namely for all generations where the proportion is between 1 and 0, but now we're going to determine the number of type A individuals using the RBinome function. For one single generation, namely the next generation, I plus 1, the population size is n as always, and then for each individual, the probability of being type A is defined by the proportion of type A present in the current generation I. Then we turn this number of type A individuals into a proportion as before and store it as the next value in the PA vector. Finally, we increment I so that we can move on to the next generation to do the same thing. So now let's execute this code in RStudio and see how it goes. So again, here's the code that we just discussed, and now we can plot it out. Now, genetic drift is typically associated with small populations where chance events have the greatest impact. To examine the impact of population size, we can modify our existing program, this R-binome method, to determine the mean number of generations until either type A or type B goes extinct out of 100 different replicate simulations for each of 6 different population sizes, namely 20, 50, 100, 200, 400, and 800. Here's one way to do so. I won't go through all of this code in detail, but essentially we have 6 different population sizes that we want to experiment with, and for each of these population sizes, we want to determine the number of generations that it takes for one type to go extinct. And we want to do this 100 times each so that we can take the average extinction time for each population size, which will give us a better representation of the situation. So now we can plot out the results, and we can see that fixation time appears to increase linearly with population size. Thanks for listening to this R-Labs tutorial video. If you found this useful, you might be interested in our free online Moodle course, or check out and subscribe to our YouTube channel.